Hello my dear students, welcome to my channel Online Biology Tutorials. This is the fourth video of the Biomolecules chapter. If you have not watched my initial videos, please do watch them before coming to this one. In this video, we will talk about the structure of proteins, and to begin with, there are four possible structures of a protein and their names are primary structure, then secondary structure, third one is tertiary structure, and fourth one is quaternary structure. Means once the proteins are formed in the cytoplasm by the process of translation, they can be in either of the structure. Okay, now we will see one by one the characteristics of each structure. To begin with, the first structure, which is primary structure, this is the easiest one. Once the protein synthesis is just over, the proteins, they are in their primary structure, right? So if we are talking about just one polypeptide, so with one polypeptide, these three structures can be possible. This is one important thing, okay? With one polypeptide. Polypeptide means a polymer which would be having many amino acids, okay? So that many amino acids will be there in a polypeptide. That polypeptide can exhibit primary structure, secondary structure or tertiary structure, fine? So the first stable structure that is formed, it is known as the primary structure. Primary structure was just the one that I discussed in my video 2 of the biomolecules chapter, wherein we saw that if amino acids are there, if 2 are combining together, then dipeptide is formed. If 3 are combining together, then tetrapeptide is formed. And if more will be combining, then a long chain, long polymer will be formed of many amino acids. So you already have an idea that when amino acids join, which bond is formed? Yes, peptide or the amide bond. So primary structure is a linear, it's a chain-like structure that tells us the sequence of amino acid, right? Here AA means amino acid. So in this linear structure, you have got an idea that how many amino acids are there? 5. Amino acid 1, 2, 3, 4, 5. Here I have written AA, but actually the amino acids will have their name. Suppose glycine, alanine, serine, lysine and methionine, they are there. So primary structure is telling us the sequence of amino acids, and it is also telling us how many peptide or amide bonds are there in the polypeptide. So primary structure is just giving us this sense, okay? What is it telling? It is telling us the sequence of amino acids, and it is also telling us about the number of peptide bonds. I hope you are clear with it. Let's move on to the secondary structure. Secondary structure would be a bit more complex than the primary structure, okay? So secondary structure is seen in two forms. It could be alpha helix or it could be beta pleated sheet, okay? Alpha helix is a right-handed helix. Means the amino acids will be there and they will form a helical structure. So this type of helical structure, it will start from N terminal, it will end at C terminal, and this is known as alpha helix. So this will also show us the number of peptide bonds. So in the secondary structure, all the forces that were there in the primary, they will definitely be there, but in secondary, some additional forces will be there. Okay, so secondary structure will give an idea on the number of peptide bonds. Of course, it will be telling us about the sequence, because once the polypeptide it is there in the primary structure, then it folds to form secondary structure. Some proteins, they are functional in their secondary structure, some are there in their tertiary structure, and some proteins are functional in their quaternary structure. So it also tells about the hydrogen bonds which are formed between the amino acids. So alpha helical structure is a right-handed coiled structure. See here, when I am moving, my hand is going towards the right side. See? And it is starting from N terminal, ending at C terminal. So this is our alpha helix, which is giving us the number of peptide bonds, the sequence of amino acids, and the hydrogen bonds that are formed between the amino acids to make this alpha helix table. Fine. Now, beta pleated sheet is a zigzag structure. It's a zigzag structure. It starts from N terminal, ends at C terminal. It will also tell us about the sequence of amino acids. It will also tell us about the number of peptide bonds. And this zigzag structure is formed because of the hydrogen bond between the amino acids. Okay. So amino acids will be there in the beta pleated sheet. There will be hydrogen bond between them. And that's why this stabilized structure is formed. So till now, primary structure we have read, secondary structure we have read. Now let's move on to the tertiary structure. And tertiary structure is very, very important. Now tertiary structure will be exhibited by a polypeptide only, and the number of forces are maximum in this. Means this is the most stabilized structure which is formed by one polypeptide. Okay, so one polypeptide, it may get coiled in number of forms to form the tertiary structure. Now the names of the forces are also very, very important. So primary structure was telling us about the peptide bonds. So these peptide bonds will definitely be a part of secondary structure. Hydrogen bonds which were seen in the secondary structure, they will also be the part of tertiary structure. Other than this, disulfide bonds will also be seen in tertiary structure. Disulfide bond means the bond that are present between the two sulfur containing amino acids. It could be cysteine-cysteine, cysteine-methionine, or methionine-methionine. So whenever sulfur containing amino acids, they come close to each other, a bond will be formed that is known as disulfide bond, and it is the strongest covalent bond. This can be asked in the MCQs that disulfide bond is a type of ionic bond or covalent bond. You need to know. Beside this, van der Waal forces are also seen. Van der Waal forces. It is VANDERWAAL. Van der Waal forces are also seen. Hydrophobic interactions are also seen in the tertiary structure. Now, very important thing, most of the enzymes in human body, they exhibit tertiary structure. You know, enzymes are the biocatalysts. They help in speeding up the rate of the reaction, and they have an active site to which a substrate binds. So this active site is formed on the enzymes by the combination of all these forces which are written here and colored yellow. Okay, so tertiary structure is the most stabilized structure of the protein. And enzymes generally exhibit tertiary structure. Their active site which binds to the substrate and leads to the formation of product, it is formed by the combination of all these forces. An example of it in your body is myoglobin protein. Okay, this myoglobin protein exhibits the tertiary structure. Let's move on to the last structure, which is quaternary structure. Now, quaternary structure is a one in which we require more than one polypeptide. Okay. So, hemoglobin is a very important example of it, and hemoglobin is a red colored pigment of the human blood. It contains an iron part and a protein part. If we see the structure of it, two polypeptides here, each line is showing a polypeptide. Okay, so by two linear lines of red color, I am showing two polypeptides and they are named as alpha. And two other polypeptides, they are similar to each other, right? So we can write the structure as alpha 2 and beta 2. So hemoglobin can be written as alpha 2 beta 2. That means two chains are similar to each other and two other chains are similar to each other. That means it is a tetrapeptide structure. Okay. So the forces that will be there in the primary, in the secondary and in the tertiary structure, they will be there in the quaternary structure as well. But the most stabilized structure of a polypeptide is the tertiary structure. Okay, very important thing, quaternary structure is formed by more than one polypeptide, and hemoglobin which is present in our body falls into its category because hemoglobin contains 4 polypeptides and they assemble together to form this proteinous molecule, which helps in carrying oxygen in our body. I hope all the structures are being understood. Now we are done with one biomolecule, that is protein. Please stay connected in the next video for the upcoming biomolecule. Thank you.